Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences at Bioengineering, IIT Guwahati, and in this module we are discussing about the different types of cells. Based on the structures, the cell could be of two different types. It could be a prokaryotic cell or the eukaryotic cell. In the previous lectures we have discussed about the prokaryotic cells. We have discussed about the structure of the prokaryotic cell and how the different components are present in the prokaryotic cell. We have discussed about the cell wall. We have discussed about the different types of analytical barriers what are present in the prokaryotic cells, and then we also discussed about the chromosomal DNA and as well as the plasmids. And in the previous lecture we have also discussed about how you can be able to isolate the plasmids from the bacterial cell and how you can be able to use them for the different types of applications, and how the plasmid is actually spreading the resistance or the different types of properties. So in today's lecture we are going to discuss about the eukaryotic cells. So when you talk about the eukaryotic cell, as I said, we are going to discuss about the two types of eukaryotic cell. One is called as the plant cell, the other one is called as the animal cell. So let's start our discussion about the eukaryotic cell. So the higher eukaryotes have the multiple organs to perform the specific functions, whereas the cell is the structural as well as the functional unit of the life, and it contains all the necessary infrastructure to perform all the functions. So besides, we have the different types of cells, like we have the prokaryotic cell, we have the eukaryotic cells, and within the eukaryotic cell you have the plant cell and the animal cells. So before getting into the detail of what are the different organelles and what are the structure of the eukaryotic cell, it is important to understand the difference between the plant cell as well as the animal cell. So the structure of the eukaryotic cell, the eukaryotic cells are much more complex and it contains many membrane bound organelles to perform the specific functions. It contains a nucleus isolated from the cytosol and enclosed in a well defined plasma membrane. So one of the classical feature is that the eukaryotic cells are actually containing the membrane bound organelles to perform the specific functions. And then you have the two different types of eukaryotic cells. You have the plant cell and you have the animal cell. So let's see what are the difference between the animal and the plant cells. These are the properties of the plant cell and these are the properties of the animal cell. So what you see here is the cell wall. The cell wall is present in the plant cell, and plant has a very robust and good cell wall which is made up of mostly the cellulose, whereas the cell wall is completely absent in the animal cell. Then the size, the plant cells are very large compared to the animal cell, and the animal cells are comparatively small. Then the chlorophylls, the chlorophyll is the light pigment, right? Chlorophyll is the light pigment what is present in the plant, and it is completely absent in the animal cell. There are exceptions where the animal cells are also having the chlorophyll, like for example the euglena, right? So euglena is animal cell, but it also contains the chlorophyll. Then we have the vacuoles. So there are large vacuoles which are present in the plant cells, and the purpose of these vacuoles are that they want to collect the food material, or sometimes they also collect the toxic substances, because the plants are much more exposed to the toxic substances, whereas in the case of the animal cell it contains the small and the small number in many number in the animal cell. Then we have the mitochondria. The mitochondria are few in the case of plant cell because the plant cell are not very motile, right? The plant cells are static, like you have seen that the trees are present in the soil, right? So they don't move around. That's why they don't require a large quantity of energy for motion, right? And that's why they don't have the very huge number of mitochondrial cells. Whereas in the animal cell you have the huge amount of animal cell, because animal cell are very motile. So because of the animal cell they are motile, they need more amount of energy, and that's why they have the more number of mitochondria. Then the lysosomes are almost absent in the plant cell, whereas they are present in the animal cell. Then we have the glycosomes. The glycosomes are present in the plant cells, but they are absent in the animal cell. Then you have the cytokinesis. Cytokinesis means the cell division. This is we are going to discuss in due course. So the cytokinesis is the cell division. Cell division is always by the plate method in the case of plant cells, whereas it is going to be by constriction in the animal cell. So these are the some of the notable differences between the animal cell as well as the plant cell. But overall, the basic property of these two cells are very same actually. They both have the plasma membrane, they both have the many of the organelles what are present. So let's start discussing about the different types of organelles what is present in the eukaryotic cell, and we are going to then take up the some of the organelles what are exclusively being present in the plant cells and so on. So let's start our discussion with the first organelle, and that organelle is called as the cytosol. Cytosol is nothing but the water or the liquid part what is present inside the cell. So you see the cell which is there, and it has a nucleus in the center, and in between whatever you see is actually called as liquid part, whatever you see is called as the cytosol. So cytosol is the liquid part filled inside the cell, and it contains the water, salt, macromolecules, which means it contains the proteins, lipids, RNA. It has an array of microtubules fiber running throughout the cytosol to give the vesicular structure to its destinations. So within the cytosol you have the different types of microtubules running, right? So these microtubules are actually making the road within this cytosol, and on these roads only the vesicles are moving, and that's how they are actually delivering their content. Just like as you are ordering the material from the Amazon, right, or Flipkart, and then the guys are coming to your place, right? But how they are coming? They are coming because there is a road, right? There is a road to your home, right? And that's how they come using these roads. So these roads are made up of the microtubules, which we are going to discuss in our subsequent lecture. And they will use these roads, and that's how these vesicles, you can imagine these vesicles are the courier guys which are coming to your home, and they are actually delivering to your place. And how they are delivering, that is a part of the very extensive vesicular structure system, and they are actually having a very well defined vesicular transport system, and vesicular transport system is actually going to help the cells to deliver the material to their destinations. Besides this, the cytosol exhibits the sol to gel transitions, and such transitions regulates the multiple biochemical and cellular processes. So sol and gel transition is actually making either the cytosol more thick or more thin, and because of that the localized sol and gel transition is actually going to make the substrates more concentrated or less concentrated, and because of that they can be able to enhance or they can be able to change the rate of these reactions very nicely. So apart from these functions, the cytosol has many of these functions. So cytosol has no well defined functions. It serves as a medium to exchange the material between the different organelles. It play in various processes, such as it is actually be a process where the signal transduction or the translation is going to take place. So cytosol has the cellular machinery which is responsible for the translation, or translation means the production of the proteins, right? So production of protein is called as the translation, and that is happening within the cytosol. Now one of the thing which people are very oftenly use, right, or interchangeably they use the term which is called as the cytosol or the cytoplasm, and there is a big difference between the cytosol and the cytoplasm, and it is very important that you should get a clarification. Cytosol is actually the liquid part, okay, so this is the liquid part, okay, whereas the cytoplasm is actually consist of the cytosol and all other organelles. So if you take the cell, okay, it has a nucleus, right? So outside this nucleus whatever you have, that is called as the cytoplasm, that contains the cytosol and it also contains the different types of organelles. So that is a very important to you understand, that you should say cytosol when you are talking about the liquid part, and you say about the cytoplasm when you are talking about the content what is outside the nucleus. So let's move on to the next organelles, and the next organelle is known as the nucleus. So nucleus is the central processing unit of the cell, and it is homologous to the processor in a typical computer. Nucleus is also called as the master of the cell because it actually regulates, it gives the instructions to the cell what it should do actually. Why it is so? Because the nucleus is containing the genetic material, and that genetic material has all the information, and that genetic material is actually going to say what the cell has to do actually. So nucleus is a very well present inside double membrane structures, right? The liquid which is filled inside the nucleus is called as the nucleoplasm, and the nucleoplasm is actually contains the different types of molecules, like so it is a viscous liquid which contains the nucleotides and the enzyme to perform the replication and the transcriptions, and as well as the DNA damage repair system, right? So the nucleus contains the nucleotides which is required for the replication as well as for the transcription, and then it has the replication machinery. Replication machinery means it is actually be going to make the DNA another copy of DNA, so that will required when the cell is going through with the cell division. Then it contains the genetic material, the DNA, in a complex fashion involving the several proteins which are called as the histone proteins. So compared to the bacterial cell, the DNA is present in a complex with the proteins, and that these proteins are called as the histone to pack into a nuclear bodies or to the chromosome. So the chromosome, when we say about the chromosome, it is a authentic chromosome what is present in the eukaryotic cell. The chromatin in the eukaryotic nucleus, so chromatin like the DNA content is present in the two forms. Either it can be euchromatin or the heterochromatin. So you can have the chromosome which is going to be either be present as the euchromatin or the heterochromatin. So euchromatin is a part of the chromatin where the DNA is loosely packed and it is transcriptionally active to form the messenger RNA. So euchromatin is actually active, whereas the heterochromatin is non-active or inactive, which means the eukaryotic cell has the mechanism so that they can be able to make some portion of their genome active and some portion of their genome inactive. Why it is so? Because they don't want to expose their whole DNA for the cellular machinery and as well as for the different types of toxicants, and because of that they can be able to protect. So the DNA what is present in the heterochromatin is actually going to be more densely packed, and because of that it is getting protected from the any kind of damages. So they will be protected from the damages, and it is transcriptionally inactive. So the portion what you require, for example, if there will be a requirement of the insulin molecule, right? For example, if we require insulin, then that insulin gene is actually going to be present in the euchromatin, and that's how the insulin is going to be transcribed within the nucleus, and that's how the insulin production is going to start in the cytosol. Since the nucleus is present inside this particular double membrane structure, it is actually well protected from the cytosol or well separated from the cytosol by the very complex structure, and that complex structure is called as the nuclear membrane. So the nucleus in eukaryotes are present in a double layer of membrane known as the nuclear envelope. So what you see here is, this is the nuclear envelope where you have the nuclear pore, and this is the only portion through which the molecules can come in or the synthesized molecule can go out. The outer membrane of the nuclear envelope is continuous with the rough endoplasmic reticulum and has the ribosome attached to it. The space between the two membranes is called as the perinuclear space. In nuclear envelope is often has the nuclear pore, and as per the calculation, an average nucleus has the 3000 to 4000 pore per nuclear envelope. So you have these type of nuclear pore, and the nuclear pore is a 100 nanometer diameter, and it contains of several types of protein. It is a gateway for the transfer of material between the nucleus as well as the cytosol. So you cannot just get anything into the nucleus, because in that is the case, there are many DNA damaging agents what are present in the cytosol. So they will not get into, because the nuclear pore is actually going to have a mechanism so that they can be able to discriminate between who will be allowed to get in and who will be allowed to go out of this DNA. The RNA formed after the transcription from the DNA within the nucleus, and it will move out from the nucleus into the cytosol to the nuclear pore. Similarly, the protein from the cytosol crosses the nuclear pore to start or initiate the replication, transcription and other processes, because you require the signal from the cell also. You require the cellular machinery from the cell so that you can be able to perform the different types of functions, for example the replication and transcriptions, and that is also being governed, or that is also being completely being controlled by the nuclear pore. Now let's move on to the next organelle, and the next organelle is called as the mitochondria, and the mitochondria is also called as the powerhouse of the cell, because it is responsible for energy production. And that's why if you recall, when we were talking about the plant versus the animal is having the more number of mitochondria because they require more energy compared to the plant cell. And as the organelle is actively involved in the generation of a molecule which is called as the ATP to run the cellular activities. Mitochondria is a double layer membrane bound organelle with the different structural properties. Outer membrane is smooth, right? So this is what you see here. This is a double membrane structure, and the outer membrane is smooth, right? So this is the outer membrane. What you see here is a smooth membrane, and it covers the complete organelle with the large number of integral protein which are known as the porins. So on this outer membrane you have the proteins which are called as the porins, and the porin allows the free movement of molecules less than 5000 Dalton within and outside the mitochondria, whereas the large molecules or the protein moves into the mitochondria through the transporter involving the signal peptides known as the mitochondrial targeting sequences. So it's not like, just like the nucleus, it's not like that anything will be allowed to get in or anything will be allowed to go out. There is a small size, like if it is a size of 5000 Dalton, that is the molecule which is going to be allowed get in through the porins. But if it is bigger than this, right, suppose you have the 50,000 Dalton, then that protein has to be go through with a well defined mechanism, which means it should have a signal peptide, so that, and that signal peptide should be of a mitochondrial targeting sequence. So don't worry about these targeting sequence and all these kind of terminologies, because that we are going to discuss. Inner membrane is folded into a membrane projection which are called as the cristae. So what you see here is the inner membrane which is being folded in the form of the cristae, and the cristae occupies major portion of the membrane and house machinery for the anaerobic oxidation as well as the electron transport chain to produce the ATP. Due to the presence of the inner and the outer membrane, the mitochondria can be divided into the two compartments. First, in between the inner and the outer membrane. So this is the compartment, right, and known as the intermembranous space, and the second inside the inner membrane known as the matrix. So this is the matrix, what you see here, right? This is the matrix. The protein present in the intermembranous space have a role in executing the programmed cell death or the apoptosis, which we are going to discuss in a subsequent modules. Matrix is the liquid part present in the innermost of the mitochondria, and it contains the ribosomes, RNA, DNA, enzymes to run the Krebs cycles and the other protein. So this portion what you see here is called as the matrix, and the matrix is actually going to have the cellular machinery. It contains the ribosome so that you can have the protein production, because you know that the mitochondria has its own mitochondrial DNA, so it can be able to do the transcription and translation, and that's how it can be able to produce the proteins. Then it also has the RNA, DNA, and it also has the enzyme for the running the Krebs cycles and all of the proteins. Mitochondrial DNA is a circular DNA. It has a full machinery to synthesize its own RNA, which means it can have the ability to synthesize messenger RNA, ribosomal RNA, and as well as the transfer RNA and the proteins. A number of differences exist between the mitochondrial DNA and the DNA what is present in the nucleus. One of the classical difference is that the mitochondrial DNA does not contain the introns. So it does not contain the intron compared to the DNA what is present in the nucleus. Now let's talk about the ATP generation machinery. So this is the ATP generation machinery which is present in the plasma membrane, and the system what produces the energy is called as the electron transport chain, which is actually a very huge complex, and that's why these are labeled as the complex 1 to complex 5, and these are all complexes are the integral proteins, and they are present in the inner membrane of the mitochondria. During the metabolic reactions such as glycolysis, Krebs cycle produces large amount of reducing equivalent in the form of the NADH2 and as well as the FADH2. Electron transport chain processes the reducing equivalent and the flow of electron through the different complexes. So what happens is that the electrons are moving from the complex 1 to the complex 4, and that causes the generation of the proton gradient across the membrane. So what happens is it is actually going to accumulate the large quantity of the proton. Then the proton expelled in the intermembranous space return back to the matrix through the complex 5, and this is what you see here, the complex 5, which is called as the ATP synthase. And what happens is that when the electrons are moving from the complex 1 to complex 4, they are throwing the proton from this side to this side, and that's how there will be accumulation of the proton. And then when this proton comes back to the matrix side, it is utilizing this complex or this ATP synthase, and because of that the ATP synthase is coupling the ADP plus Pi to generate the ATP, and that is because ATP synthase is a very big complex. It has the F1 unit and as well as the FO unit, and so ATP synthase is a mushroom shaped multimeric protein complex mainly composed of two proteins, FO and F1. FO is a membrane bound portion, so this is the FO, right, which is a membrane bound portion, and F1 is the complex present into the lumen towards the matrix. FO-F1 complex of mitochondria harvest the protein motive forces to catalyze the phosphorylation reaction responsible for generating the ATP. Now what are the different reactions, what are the different function of the mitochondria? So the first and the most important function of the mitochondria is that it is involved in the ATP productions. Then the second is it is actually be responsible for the generation of reactive species to kill the infectious organisms. So when there is the entry of the infectious organism within the mitochondria or within the cell, it actually induces the production of the reactive species. Reactive oxygen species means any molecule which actually contains the single electron. So because of this single electron, they are very toxic, right? So because they are toxic, they are actually being used as a weapon to kill the microorganisms. Then the mitochondria is used to track the trait of a family. You know that the mitochondria, if you see the production of or the how the sexual reproduction occurs, right? During the sexual reproduction, you have the two species, right? You have the sperm, right, and you have the ovum. Ovum is actually a complete cell. It contains all other organelles, so it contains the mitochondria also, whereas the sperm is only containing all other organelles, but it only contains the DNA. So when there is a fusion of the sperm and ovum, it takes the DNA part from the sperm, whereas it contains the all other organelles from the ovum. So because of that, when the zygote is being developed, it actually contains the mitochondria, right, and that mitochondria is from the mother side, okay. that mitochondria remain constant, because even if this particular individual will again go for the sexual reproduction, that mitochondria is actually going to carry forward, and that's why you can be able to use the mitochondria to track the family tree. Then the mitochondria is also having a role in the programmed cell death or the apoptosis. So apoptosis is what we are going to discuss in the subsequent modules, but I have given a very short write up so that it will actually help you to understand what is apoptosis. So apoptosis is a programmed cell death involving the series of events involving cellular metalloprotease known as caspases in an adverse event of exposure of the cell to the cytotoxic agent or to the environmental conditions. It activates the cell surface signaling to activate the cytosolic proteases. In addition, it disturbs the mitochondrial membrane potential to cause the release of a protein which is called as the cytochrome C. As you remember, right, when we were talking about the transport of material within the cytosol and outside, we said that the porine is only allowing the 5000 Dalton proteins, right? But if there will be a membrane potential problems, right, if there will be a drop in the potential, then that will allow the leakage of the proteinaceous substances from the mitochondria, and one of such protein is called as the cytochrome C. And once the cytochrome C is going to be released from the mitochondria, it is actually going to activate the different types of DNases, like, and that's how the DNAs are actually going to destroy the DNA what is present inside the nucleus, and ultimately it is going to destroy that particular cell. And this is actually a very programmed death, and that's why this is called as the apoptosis. Now let's understand the first process, how the mitochondria is actually synthesizing the ATP. So to understand and to explain this process, the people have put forward a theory which is called as the Chemiosmotic hypothesis. So what it says is that the F1 particle, so you remember, right, it has two particles, one is FO particle, other one is F1 particle. FO is the integral membrane protein which is only responsible for holding the F1, and it also gives the path for the protons to enter, right? Whereas the F1 is actually a multimeric protein complex, and it contains the three different proteins. It contains a protein which is called alpha subunit, it contains the beta subunit, and it contains a gamma subunit. So this is the gamma subunit what is present in the center, and the function of the gamma subunit is that it actually only transfers the complexes from the alpha to beta. So what you see here is that these alpha and beta proteins or the subunits could be present in two different conformations. Either it could be present in the loose binding conformations or the tight binding conformations. So when they are present in the tight bind conformations, they will bind that particular molecule very tightly. So what will happen in the beginning? In the beginning, the ADP and the PI is actually going to come and bind to the loose binding site. So once they will bind to the loose binding site, the gamma subunit is actually going to take these molecules and put it into the another subunit. And once they will reach to another subunit, the ADP and PI are actually going to mixed with each other, and that's how they are actually going to form the ATP. As soon as it forms the ATP, it will again be flipped to another subunit, and that subunit is open complexes, and that's how it is actually going to it is going to release the ADP, and then it is actually going to take up the new molecules of ADP and PI. So this cyclic event is going to continue for many rounds, and that's how it is actually going to keep giving you the ATP synthesis. So I have requested one of the professor in MRC, and based on my request, I think he has shared a movie which actually explains these processes very nicely. So Professor Walker, we should be very grateful to him that he has shared this very good animated schemes. So in this scheme, they have shown how the ATP synthase is actually synthesizing the ATP utilizing the different types of complexes. So what you see here in this movie, yeah, so this is what you see here. This is the membrane bound structures, and this is what you have an integral protein. So this is a, yeah, so what you see here is this cyan color portion is actually the FO particle, and all these what you see here is F1 particle. This is what you see is the gamma chain, whereas this blue yellow is the alpha chain and this is the beta chain. So what when you will see that when it is actually utilizing the proton motive force, it is actually rotating, and when it is rotating, it is actually accepting the ADP and PI, which is in the loose binding site, and once the loose binding site is rotating, it is converting into a tight binding site, and that's how the ADP and PI are mixed together and it generates the ATP. Again there is a rotation, and because of that it is getting converted into the loose binding site, and that's how the ATP which is formed in the previous cycle is going to be released. So this is a very good movie to understand the whole process, and that's how you will be able to appreciate how these events are actually been happening. So what you see here is actually it is showing how the ADP and PI are combining with each other. So you see these are different bonds through which the ADP is combining with the PI, and that's how the ATP is formed, and that's how it is actually being released from the system. So this is all about the ATP synthesis and its mechanism by the ATP synthase. And what we have discussed so far, we have discussed about the eukaryotic cell, and within the eukaryotic cell we have discussed about the differences of the eukaryotic cell with the plant cells, and then we have discussed about the nucleus, we have discussed about the cytosol, and we have also discussed about the mitochondria. Now in our subsequent lecture we are going to discuss about some more organelles from the eukaryotic cell, and then we also going to discuss some more interesting aspects related to the cell. So with this I would like to conclude my lecture here. Thank you.